Hi, my name is Sarai M. Rivera Rivera. I am from the University of Puerto Rico at Arecibo, and in this summer I decided to do a summer research project at Michigan State University in the Department of Physiology. I had the opportunity to work in Dr. Eran Andrashek's lab with his research project about a specific PTPRH mutation that results in EGFR pathway activation. Lung cancer is the leading cause of deaths in the United States and worldwide for both men and women. Cancer can start anywhere at the body, and the main problem with this disease is the uncontrollable growth of cells invading other parts of the body. The fact that 5% of lung cancer patients have a protein tyrosine phosphatase receptor type mutation suggests that certain PTPRH mutations cause activation of epidermal growth factor receptor EGFR, with a fraction having EGFR translocates to the nucleus, changing gene expression. This PTPRH phosphatase oversees the phosphorylation of EGFR, a known driver of tumorigenesis. The patients with these mutations present a pressing need in cancer biology since they are not given an EGFR-targeted therapy and instead suffer from the side effects of chemotherapy. Early work from Dr. Andrashek's lab shows that the PTPRH mutation results in high levels of phosphorylation of EGFR, causing unwanted cell growth in mouse. Our hypothesis states that PTPRH mutation is causing that EGFR translocates to the nucleus, changing gene expression, and that these mutations arise early in tumor development. To prove this hypothesis, we sequenced the DNA from mammary glands of 21-day polyomavirus middle T antigen mouse and mammary glands of 35-day PYMT mouse. By extracting the DNA, running PCR of the stretch covering the B483 mutation of PTPRH, agarose gel, and send them for Sanger sequencing. This is an example of a typical tumor. I extracted DNA from mammary gland of wild type and PYMT mouse. The fat pad is pink and surrounding everything. The orange arrow is pointing to the lymph node, the blue one to the ducts, and the black one to the terminal end buds. They carry the milk back to the nipple. In the 35-day polyomavirus middle T antigen mammary gland, the red arrow is pointing to the hyperplasia. The mammary epithelial cells here express high levels of PYMT, and because of this expression, they get a hyperplasia, overgrowth, which is not yet a tumor, but it does progress to a tumor by around average of 45 days. 50% of the sequence results from the 21-day PYMT mouse presented the PTPRH mutation. In here, you can see a substitution of the DNA base pair. Instead of a C, a T. On the bottom, there's an example of a wild type. In the 35-day PYMT sequence, you can see the same substitution. We know that we can treat EGFR with tyrosine kinase inhibitors. There are papers that show effective treatment with these inhibitors against EGFR. We believe that we can link all these things together. Polyomavirus middle T antigen is a driver of tumor formation. We were surprised when we found PTPRH mutation in the 21-day PYMT samples before getting tumors. We got a mutation in PTPRH, which means EGFR is on. My work shows that this mutation is driving the cancer. The fact that these mice at day 21 of development do not have a tumor yet, but we found that mutation in the mammary gland is proof that this is the reason that these mice are getting tumors. If we think about human cancer, this is a key step for developing human cancer too. We are waiting for the approval of the Institutional Review Board to work with human samples. Once we get this approval, we will repeat DNA extraction, PCR, electrophoresis, and Sanger sequencing with 250 non-small cell lung cancer human samples. I would like to give special thanks to my PI, Dr. Eran Andrashek, Ashley Perry, Carson Broker, John Busich, and NIESS Grant for letting me have this amazing opportunity.